Good morning, students. Welcome back to lecture 6. Today we'll be talking about antibodies. In the last class, we talked about antigens, but in general, I gave overall introduction of proteins, structural units of fundamental units of proteins, amino acid, different conformational changes proteins go, primary, secondary, tertiary, and quaternary structures, amino acid residues, different bonds, hydrophobic interactions that can exist among proteins. That is very important to understand structure and function relationships. At the same time, in this today's lecture also, we are talking antibodies. Remember that antibodies are also protein molecules. So just we'll quickly go through whenever that information comes, but I have given enough fundamentals in the last class. I will presume that we are on the same page and we can go quickly today's lecture. So the outline of the lecture is divided into 5 to 6 concepts. The first one is antibodies. You know that they are also called immunoglobulins. Why they are called immunoglobulins, we will see in this class. And structure and properties of antibodies, and types and subtypes of immunoglobulins or antibodies, main functions of antibodies, and then generation of immunological diversity at the genetic control, or looking into the molecular biology of which are the genes coding for specific types of subtypes and types of immunoglobulins. But I think that will be covered in the next class, but depending on the time, I will try to cover as much as the top 4 concepts in this class. The antibodies. In just general view, we know that antibodies, they bind to foreign substances that invade the body, such as pathogens. Why do you call anti and body? Because the body that invade the body are pathogens, foreign bodies, that's part of the body. Whatever that attacks the bodies, foreign bodies, it's called anti and body, antibody. That's a term refers to its function in general, which is to bind to antigen. Another name of this protein molecule is called immunoglobulin, abbreviated as Ig. Coming on to recap of previous lectures, antibodies produced by B cells, mainly B plasma cells, through humoral immunity. They are membrane bound. They are membrane bound on B plasma cells. At the membranes, antigen interacts with the antibody to elicit immune response that gives a clonal propagation of B cells having a studied in the previous class. Antibodies are secreted. The secreted antibodies go into the blood flow and they encounter the antigen and neutralizes them. We will see briefly how they does it in this class. And they have a structure in common. Antibodies have a structure. They have an ability to recognize and bind antigens, and they can share a specific effector functions common to immune cells. What we will see. Why are the antibodies called immunoglobulins? So if you divide the term on immunoglobulin, you cut it into immuno and globulin. A globulin having an immune function. What is globulin? Again, globulins are a specific set of protein molecules, class of protein molecules. We'll see why the name immunoglobulin. You already remember in fundamental classes in bachelor that you talk a lot about vaccination, antibodies, rabbits, the other animals, antisera was raised for specific antibodies. We specifically talk about a term called serum antibodies. The serum antibodies are usually the location of the antibodies present in the blood. It's called as a serum part of the blood. That's why they call it as a serum antibody, because when you are doing an experiment to isolate antibodies from the blood samples, from the blood, you are looking into a specific portion of the blood called serum. How the serum is formed? You see into this kind of test tubes here that contain the blood. And you remember that blood has a fluid fraction and cellular fraction. For example, you take certain ml of the blood, you go by a process of centrifugation by mass, the particle sediment, right? If you take here a sample of the blood, normal blood, and also you remember the factor coagulation, clotting factors of the blood, fibrinogen and fibrin. So a normal blood that is not coagulated, not in the form of coagulation, consists of these parts when it's centrifuged. The heavy part are called red blood cells. They go into the bottom. And the supernatant is called plasma cells. It contains fibrinogen and fibrin molecule. In between, there is a buffy coat made up of white cell cells and platelets. In terms, you see what is the fluid fraction, cellular fraction. These are the cells representing normal. Let's say a factor that induces clotting, then the fibrinogen will not work properly, and the fibrin molecule will cross-link the blood, specifically the plasma. And when you do the sedimentation for the clot, all the cells will sediment into the clot here, the red portion. Remaining portion stays in here of the plasma and other portion, other cells. Those are called as serum. This serum in the clotted. So it came to know that when you do electrophoresis, you separate this blood, take the blood samples, take the clotted blood samples, undergo electrophoresis to identify and separate different proteins present in that, different fractions, fraction from the clot, fraction from the serum. They were able to identify that most of the antibodies are present in the serum. That's why they call serum antibodies. How they were able to identify, we'll see in this experiment why they are called globulins. We'll see in the experiment that explained in the textbook Kube Immunology, chapter 4. This experiment was done by Tiselius and Kabat in 1939. See the timing. Because at the time electrophoresis was, gel electrophoresis, agarose and protein electrophoresis was going good. So they were able to understand. So this graph will tell you that why the globulins are majority of the family of proteins that go and recognize antigens compared to alpha, beta, or albumin member of the proteins present in the blood. So what they have done in experiment is here, you see one light blue or blue and then black arrow, the black graph that represents two different conditions, black and blue. So just to go explain on this, remember that so when you, for example, when an antigen encounters for the first time, the immune system recognizes it, it produces antibodies, right? Then clonal propagation happens and a specific set of antibodies go and bind a specific antigen. So basically, given the pool of the blood, given the pool of lymphoid organs, if you want to identify a specific antibody for a specific antigen, you can go with protein-protein interaction. You send a specific antigen to a body, into the blood, or immunize a specific species, and then see that whatever the antigen, capture the antigen after certain immune response and go for immunofluorescence, go for electrophoresis. You can find different kind of protein that are attached to the antigen. So for example, specific set of antibody that attach to the antigen, you can pull it through antigen-antibody complex out from the system and then analyze that what are the antibodies specifically catching to the antigen of the given pool of proteins, pool of antibodies. So what these people have done in experiment is that, so after rabbits were immunized with ovalbumin, it's an antigen. Rabbits, they don't have this protein in their system, it's an antigen for them. Then they raised anti-sera from that rabbit blood, they raised anti-serum, collected the serum. And then they divided the serum into two portions. One portion represented by this blue, another portion represented by this black, and they did electrophoresis experiments. Positive to negative proteins go with the charge, they go with that. And they separated two pools. One is different into two pools. Let's say the pool is blue and the pool is black. What they have done, pool A, pool B, but the pool A that is blue, they kept as it is. So they know that the ovalbumin has elicited immune response. They have given specific pool of proteins or antibodies in the body. They kept it as it is. They electrophorese it. And they see that of these specific pools, there is kind of albumin high, but in the terms of globulin, alpha, beta, and gamma are there. This is just the initial response of the antigen, and they pooled it. Then our remaining portion B of the same antiserum A is blue, antiserum B, what they have done, they again treated it with ovalbumin, or they want to isolate whatever the antibodies bound to the ova. So they incubated them with ova, that is antigen, to remove anti-ova antibodies, whatever attached to this. So after response of that, in the black, what they have done, the electrophoresis, by a condition, they removed whatever that antibodies are present in that by antigen-antibody interaction. Then they again electrophoresis the antiserum part B after removing the antibodies bound to the antigen. Okay, is it clear? So what they understood after that, specifically of this category of proteins, only the gamma albumins, the pool of gamma albumins in that part B of the antiserum got drastically reduced because they removed it, and they know that by this reduction in the proportion of the antibodies present in the pooled and unpooled from the antigen, taken out from the antigen antibody complex, they know that a specific portion of gamma globulin specifically reduced, saying that these are the majority that went and bound to the OVA, that is the antigen. Okay? So there may be, excuse me, there was a slight reduction in alpha and beta globulin also, but it is not significant compared to gamma, and there is no change in the albumin at all. Either you pull it out or not. In those both conditions, part A and part B of the antisera, the albumin did not attach to the antigen OVA, only the alpha globulin attached. This is a fundamental experiment. It is really important for you if you want to become an immunologist. To try to understand how do you pull a specific antibodies giving a specific antigen, how do you do electrophoresis. These are the best techniques you need to learn if you want to go in a clinical immunology lab to work as a research assistant or scientist. Okay, coming on to epitopes and antibodies again. It's a recap of the last class. Remember that a pathogen elicits immune response to produce antibodies that are heterogeneous in nature. What I'm saying is that a pathogen, a bacterial cell here, elicits immune response to produce multiple diverse antibodies which are varied from each other. Why they do that? Because bacterial cell, a pathogen will have several antigens on the surface, cellular components or cell wall components, several antigens, and then when you zoom into the antigen level, each antigen we studied in the last class that will have different antigenic determinants called epitopes. So a pathogen will have multiple antigens on the body, and each antigen, when you zoom in into single, each antigen will have multiple epitopes again, and then each epitope will bind to a specific antibody. Again, you are seeing a singular here, you are seeing a plural here. Pathogen to antigens plural, antigens to epitopes plural, but epitope to antibody singular, that is a specific interaction happens here. But when you see a pathogen to antibodies, again it's plural. You get heterogeneous in nature, several antibodies will come up. So again, how the antibodies will bind the antigens based on the variable regions, you've seen that. We'll look in the next slides, antibody A, antibody B. And this is the kind of chemical structure going into zooming of these antigens here. Each antigen will have a chemical structure, chemical conformation, chemical beta and alpha sheets, tertiary quaternary conformation, and every epitope, that is functionally different chemical units along the protein, that is antigen, will have diverse variability. Of this diversity, you will get a diverse pool of specific antigen binding in each region. So again, the response, this is the basic again what I explained in the previous slide. A response, immune response that recruits clones of B cells, and their output is in monoclonal antibodies. Each will bind to a single antigenic determinants. It goes into heterogeneous population, polyclonal antibodies, it immunizes specific antigen. Coming on to structure of antibodies, also this is very important. You may get a short and 2 mark questions on what is heavy chain and light chain. We will take a little bit more time, try to understand the structure, basic structure of the antibodies. So you're looking like 4 lines here, right? 1, 2, 3, 4. 4 lines. Each line is a peptide. That is 4 peptide chains. In fundamentally, antibody, any antibody, given an antibody, the structure is heterodimer. It is a heterodimer and it has both heavy chain and then light chain. So heavy chain here, light chain here. Why they are saying heavy and light? Based on the size. Light chain is that is 25,000 daltons. Heavy chain is equal to 50,000 daltons or more. Why they are more said? Because they have more amino acids in terms of short and long length. You see that this is a short chain, light chain that has only 214 amino acids composed of amino acid units, individual units. It's a polymer, polypeptide. And then heavy chain consists of 446 amino acids. Accordingly, the size changes. And when you see the number, we have 2 light and 2 heavy. That is called dimer of dimers. 1 heavy chain, 1 light chain, 1 heavy chain, light chain, dimer of dimer. That consists of 2 and 2, 2 plus 2, it's a 4 peptide chains. That is the fundamental structure. Again, in between, each chain will have separate structural uniformity, structural heterogeneity, and kind of cross-linking between that. You see, in terms of here, a heavy chain can interlink based on a disulfide bond. It can interlink with a light chain by a disulfide bond again. In between, within these each individual chains, they may link by peptide bonds. Amino acid binds to amino acid, and also based on the light and heavy chain, there is variability of based on the some minor variabilities of this light and heavy chain. The antibodies are divided into different classes. For example, the light chain will always have either K or so gamma here, K or gamma, specific light chain variants. Only 2 variants are available in the light chain, but the heavy chain, it will have 5 variants based on these symbols, mu, epsilon, psi, alpha. You can see into that gamma here. So you can change based on this variability of heavy chain. You call them as different variabilities. But remember that these, and also, for example, I will talk here again, it's okay. But coming on to these complementarity determining regions, we know that the variable regions VHVL, VHVL, these are the regions that bind and attach to specific epitopes and the antigen determinant regions. They will have a complementarity in terms of structural bonds. That's why it's called complementarity determining region. CDRs specifically present in the variable regions, that is the antibody binding. And the other regions is called constant regions. These have other biological activity. We will see that. This is only antigen binding. After binding the antigen, how they kind of kill the antigens is how they biologically regulate the processes. This activated by these constant regions, because these constant regions will have receptors that binds to specific macrophages. We will look into the next few slides. And then FAB fragment is antigen binding. FC fragment is called, usually this is FAB, this is called FC. We will see in the next slide. Crystallizable, this is the antigen bond. Remember that the heavy chain has 5 variables. Of the 3, they will have a hinge region common here in the middle. But the 2 of these classes, mu and epsilon, they don't have the hinge region. Like disulfide bond will be here. These 2, the black thing here. They don't have it here. Based on the structural variability. Briefly read it into the text here. Try to understand what I explained briefly. So again, this FAB, antigen binding, FC is a constant region. A very beautiful diagrammatic sketch by Thermo Fisher. You can go through again this. Remember that I spoke about biological activity. This is an antigen binding site, antigen recognition activity here. But the FC has biological activity. The biological activity is concerned with a specific post-translational modification on the constant region of the antibodies. That's called glycosylation. Glycosylation, because a specific oligosaccharide sugar can go and bind here, not here. Binds in the constant region, and that's why immunoglobulins are also called glycoproteins, a combination of protein and then sugar. Coming on to different types of antibodies. In general, we know that common to our species, common to any species, human species taken out, there are 5 types of antibodies, keeping the common structurality. There are 5 types. Those are called by a mnemonic, you can remember that, GAMED, G-A-M-E-D, GAMED, which represents a specific symbol of the antibodies. See, gamma, alpha, mu, epsilon, and then D is delta. So these are the symbols here. You can remember this mnemonic called IgG, IgA, IgM, IgE, and IgD. These are the common antibodies present in a species. And fundamentally, they differ in structure in terms of units present. We will see that, for example, in IgM, we know that this is fundamental Y-shaped unit, right? Some of these antibodies will have only 1 unit. They may differ in the size and length, but some of them will have a dimer, some of them will have pentamer. For example, IgM is a pentamer forming of these Y units. IgA is a dimer formed of this unit. Okay. These Y units, the valency will changes. The monomer, give a second, please. My daughter is kind of recording. Okay, so the antibodies differ by the valency of the Y-shaped units. And then, for example, you see here the difference, IgA is a dimer. The link between two Y-shaped link is more attached by a unit called J chain. What is the J chain? You can see again one more summary of the slides that can give different types, different properties of the antibodies. You can see in this slide what we talked, because based on the molecular weight here, you can differentiate them. Based on the percentage of antibodies present, different types of antibodies present in the antibody in the serum, you can see that common is IgG, 80%, most abundant, most less is IgE. And based on the function, you can see that they have varied functions. Some of them are in the primary response, some of them are in secondary response, some of them specifically interact with the parasites, some are secreted mainly, some follows a specific process called opsonization. Fixes complement, yes, yes, no, no, no. What is complement? We will see in the end of the class. So this is important. You can remember the different function, structural aspects of the antibodies. You may be asked in the exam some of these points. Again, how the variability happens in terms of this, why there are different 5 types of antibodies, mainly because though they have a constant heavy chain, constant FC region, and then variable antibody region. In the FC region also, they have a differences in the polypeptides, specifically amino acid sequences. There is a kind of variability, polymorphism, that causes them to do different immune response, different to be present in a particular stages of immune response, and then do a particular different immune response. The polypeptide protein sequences responsible for they are found in the FC fragment, constant fragment, not in the antigenic determinant fragment. So the J chain is nothing but a protein component of the antibodies IgM and IgA. It is a 137 residue polypeptide encoded by a gene IgJ gene. You see briefly on Ig chain here. Again, the 5 types of antibodies are divided into several subtypes based on the changes in the subclasses. For example, here IgG, heavy chain is gamma, light chain may be KR alpha. KR, these only 2 types of light chains are there. These symbols represent different that gamma and K and other symbol here. So the light chain, heavy chain, but the subclasses based on the heavy chain variability again in the heavy chain, gamma 1, gamma 2, gamma 3, gamma 4. Again, there is variability. 5 IgG antibodies that differ individually based on the heavy chain. Taken an IgG of a different species, for example, my IgG and your IgG, it may differ within the heavy chain again. Okay, so 4 types here, the formula can be here. So remember that based on the heavy chain, as humans, we have 5 different types of antibodies. Again, between species of humans, based on the changes in the heavy chain, again, individual heavy chain, we might have subclasses. We call them isotopes, allotopes, and then idiotopes. We'll see in the end of the class. So in the next few slides, just they will speak about the fundamentals of protein-protein interaction, protein structural conformation that we covered in the last class. Just now you are not looking in the antigens, you are looking in the antibodies, how the conformational changes happen, how the primary, secondary, tertiary, and quaternary structure of antibodies will happen. We are taking an example of here, immunoglobulin fold structure. For example, one immunoglobulin you are seeing here, the constant light chain, variable light chain, variable light domain in terms of you're talking in the constant light chain domain, variable light chain domain, you're looking into a kind of quaternary structure of the protein. This protein here in this domain, CL, constant domain, variable domain, they have a specific loops of amino acids formed by amino acids, and they have a specific beta strands or beta sheets formed by quaternary, it's a secondary structure, right? Beta sheets or beta strands, secondary structure organization. Each of them will again disulfide bonds, amino acids in between them peptide bonds. There may be loops along this structure. And then how the beta strands are arranged, they are parallel, anti-parallel beta strands exist. In this specific case, they are anti-parallel here, both of them combined by disulfide bonds in the constant domain here, constant long domain here. In the variable domain, you are seeing a specific blue structure here. It's not there in this constant or FC. This is FAB or VL domain. You see these blue regions, that is a specific out of these B sheets. They are called CDRs, complementary determining regions or antigenic determining regions. and they will be variable based from antibody to antibody type, antibody subtypes. And also you see that coming to the beta sheets here, 4 plus 3, here 4 plus 3, and 2 more extra beta sheets are there in the variable domain. So beta sheet, pleated sheets, anti-parallel, disulfide, light chain differs with V chain. There are 2 extra beta sheets here. And then hypervariable loops, these are called, they are variable antigen determinants. Similar sites are also present in the heavy chains are also. You're talking about the short and long chains, you may see specific regions in that also. How the variability you determine? Determined by different amino acids present at a given position within these secondary or tertiary structures, and the frequency of common amino acid in the given position. You might have a common amino acid residues, variable amino acid residues, variability of antigen, antibody production, you can check on this formula. Again, coming on to quaternary structure. So the beta sheets, amino acid residue, disulfide bonds, they go into one more conformational change called quaternary change. The quaternary conformation change mainly works on hydrophilic hydrophobic interaction, how these molecules fit themselves in space in three-dimensional structure. So this is one of the excellent crystallographic of IgG antibody. They have a specific Y-shaped structure, specific domains in three-dimensional conformation they are placed. And then you see that one of these CH2 domain is the one that binds to the sugar moiety, not the antigenic determining region. Here is the carbohydrate. And remember that binding to a specific antigen or specific protein molecule, protein-protein interaction, a specific three-dimensional protein X can interact with three-dimensional protein Y. Binding of this can give a conformational change, that the conformational change gives a specific unique function. For example, you are seeing here, you're seeing specifically see here an antigen bound to an antibody. The antibody is represented by 2 colors, 2 shades, blue and pink. The blue may be in the conformation. The red line shows the structure of FAB fragment before it binds to the peptide. The red region is that in 3-dimensional, a kind of cleft antigen is reacting with the antibody. Before binding to that specific active site interaction, there is a red conformation. After binding, the conformation shifts to a blue that will be recognized by extra crystallography. That changing of conformation is important for the structure of the antigen antibody. That antigen antibody interaction brings protein-protein interaction. So what is the biological activity of antibodies? How this conformational change elicit a biological function of immune responses, specifically for antibodies? We will see in the next few slides. Specifically, antibodies have effector functions in the immune cells, 3 processes. One process is called opsonization, that is mainly promoted by antibodies, like by binding to a specific antigen. It goes through a biological activity of killing specific antigens or foreign bodies by a process called opsonization. Antibodies activate a complement system. What we will see, what is that complement, what does it do? And then antibody-dependent cell-mediated toxicity, ADCC, is a specific process that kills cells. This process happens mainly in interferons, mainly cytokines are produced, cytotoxins are produced, specifically for virus or some other pathogen that we will see. We may not cover in the present class these 2 concepts. We will cover in then coming to the next class, complement system. And the ADCC can be covered in specific class where we talk more about interferons and cytokines. But we will briefly see opsonization. Opsonization is the process whereby opsonins make an invading organism more susceptible to phagocytosis. Nothing but a ligand-coated particle, an antigen, will be coated by specific molecule called opsonins. By doing this opsonin binding, specific phagocyte can recognize this antigen and enhance the phagocytosis by a specific receptor present on the phagocytes. These are opsonins that bound to the antigens here. So major opsonins are immunoglobulins. IgG antibodies are also act as opsonins. They bound to the antigen antibody, specific IgG antibody bound to an antibody elicits a phagocytosis process. Specific cells will bind that. There is one more certain plasma lectin called mannose binding lectin that is also act as opsonin. And there is a complement system C3b that is also acts as optosin. All these molecules present an antigen. They trigger a process of phagocytosis. The phagocytosis is done by specific dendritic cell are antibody binding cells here. Macrophages and neutrophils, they elicit phagocytosis. These molecules will have receptors called Fc receptors. These receptor can recognize specifically the opsonins present on the antigens. This, for example, opsonin, what is opson? It's coming from a Greek word. The beginning of the class, I was speaking about the desserts, what homemade desserts are you are enjoying or savoring them. In the Greek word, the option is nothing but a delicious side dish of any meal in a Greek word. And it is comes into the normal meal that is called cetos. Along with that, a dessert, you will like to savor the dessert. Same way, the phagocytes, they would like to, along with the antigen as a meal, cetos, they want to see these opsons as a dessert molecule. It will enhance the taste buds. Basically, the more phagocytosis will happen fast enough, so that these opsons, they just treated as a dessert molecule. So specific complement system, there are serum proteins, glycoproteins, they're called complement system. Specific protein molecules are there in the immune systems, mainly in the serum. These are called as mainly how the complement system works is that basically it perforates the cell. Any cell that has attached to the complements, it causes a perforation in the cell membranes, basically killing the cell. So how this process and mechanism happens, we will see in the next class. But any complement, any molecule, any foreign pathogen that attach to a complement, they will have a specific fragment called C3b. Attachment to this, they will activate a process of perforation. How the complement system kills that. Again, IgG is mainly involved in the complement system. We will talk in the next class. Again, antibody cell-mediated toxicity, specific cytokines or toxins are reduced. For example, interferons recognized. Special natural killer cells will kill that. That also we will see in the further classes. This brings to abbreviation of isotypes, allotypes, and idiotypes. Already we spoken that there are 5 classes of immunoglobulins. Why there are 5 different types? Because they vary slightly in these heavy chains here, in the dark here. IgG, IgM, they have different units on this that. Structural variation causes different isotopes. Of these isotopes, my IgG will differ from your IgG. For example, strain A, strain B, IgG1, IgG2. They may different again in the heavy chain region, constant region. And again, here we represent one unit of change. Here we represent multiple unit of change in the constant region. The variability gives rise to allotypes. Allotypes are nothing but common to a different species, but variable from strain to strain. And idiotypes are based on the antigenic determinant. You are seeing the black shift here, not the changes in the antigen determinant region. So you go and look into the abbreviation, you see more different kind of terminology here. You go and write notes on that. That will help in the 2-mark questions. So we have less than 1 minute or 1.5 minutes. Quickly, I will give how we can generate different antibodies in the body. For example, if you don't elicit any immune response in a system also, and constantly, we might have 10 power 12 different antibodies in the given system. You don't need to give antigen stimulation, premature antibody repertoire. You will have 10 power different antibody molecules. But the maturity happens, there is enhancement pool of that. There will be antibody arsenal will come up, millions and billions of cells after maturity. You look into that. How it happens? Because an animal with 100 genes encoding light chain, 100 genes encoding heavy chains in principle can produce 100 in 1000 into 1000, 1000 into 1000, 10 power 6 different antigen binding sites. How it will happen? By combinatorial gene expression, combinatorial genes, the junctional diversification and combinatorial of L and H chains that we will speak in terms of genetic fundamentals of genetic response in the next class, how antigen variability can come up. Okay, I would like to stop the class here. Variability of antibodies, how a repertoire can produce, we will see in the further class. Okay, thank you.
Thank you, sir.